Introduction to molecular basis of inheritance. A very common and important terminology, which is gene. Let's discuss. Unit of heredity and is responsible for inheritance. So in brief, gene is defined as the structural and functional unit of heredity. Gene is that part of chromosome which is made up of nucleotides. It grows, reproduces and mutates and is thus considered as the ultimate unit of recombination. Now, gene, as it is said, it is the unit of heredity and also responsible for inheritance. It is like when transmission of characters pass from parents to the offspring, it is the genes which is carried forward, carrying all the desired information from the parent to the offspring, or it is the unit which transmits generation to generation. That is why it is said that gene is the unit of heredity and is responsible for inheritance. Gene is the structural and functional unit of heredity. Just like cell is the structural and functional unit of life, so the base for heredity, which is the structure and functional basis of heredity, is the gene. So gene is that part of DNA. So gene is a part of DNA and is made up of nucleotides. So if this is the DNA, let's consider this as the DNA. So far we have not discussed what is DNA in my earlier videos, but let's talk about DNA. DNA is deoxyribose nucleic acid. So DNA is made up of number of nucleotides. So gene is a part of the DNA and is made up of nucleotides. Suppose take this as DNA and this part, let's show with the red color. This part can be a gene. It is a part of DNA and is made up of nucleotides. So as the DNA, we will discuss what is DNA in my next videos. As the DNA reproduces, mutates and grows, likewise gene, as it is a part of DNA, it grows, reproduces and mutates by itself, and thereby it is considered as the ultimate unit of recombination. So, central dogma of molecular biology is a very important and commonly used term sentence, that is central dogma of molecular biology. Sir Johansson coined the term gene as the heredity factor of Mendel. So Mendel, he stated that there is something called a hereditary factor which transmits generation to generation. But Sir Johansson coined this hereditary factor of Mendel as gene. And the central dogma states that DNA to RNA, RNA to protein. It is the information which is transmitted from DNA to RNA by a process called transcription, and then the information from RNA is translated to desired protein. So the central dogma of molecular biology is DNA to RNA, RNA to protein. I will discuss this in detail in my next videos. DNA to RNA, as I said, is converted by a process called as transcription. Before that, DNA replicates by itself. This is another term. So replication is an important terminology, then transcription, then DNA replicates first, then it transcribed to RNA, messenger RNA, and then translated to desired protein. This is altogether the central dogma of molecular biology. Very interesting terminologies: transcription, translation, replication. I will discuss in details in my next video. So, is defined as unit of recombination, mutation and self-reproduction. So, gene is the unit of recombination, mutation and self-reproduction, means it can reproduce or replicate by itself. Gene is a macromolecule attached to protein and can pass from one cell to another and can be transmitted from one generation to another. This I have mentioned earlier. Very important is gene is a macromolecule which is attached to a protein. Now what is genome? It is the total number, this terminology is very important, genome. It is the total number of genes present in haploid set of chromosomes. So genome is the total number of genes present in haploid set of chromosomes. Number of genes in one set of chromosomes ranges between 10,000 to 15,000. Approximately total number of genes in man is about 3 lakh. Genes contain DNA as primary component. Locus, what is locus? Each gene has a specific position in a specific chromosome. That specific position of where the gene is present or located in a specific chromosome, like a beads on a string at a fixed distance, is termed as locus. It is like this: beads on a string. So these are genes. Now the properties of genes: duplication by replication. Gene can replicate double in number and equal distribution when it is divided from parent to the next offspring. So gene is duplicated and distributes itself equally, and it is able to undergo mutation by itself. Mutation, expressing the genetic information by transcription to mRNA, as I mentioned in central dogma. That is, gene is transcribed to mRNA to produce proteins. Chemically, genes are made up of DNA. Genes produce proteins, an important protein which is an enzyme. Proteins are made up of the polypeptide chains of amino acids. Now what is protein? I will discuss in my next video. Protein in biochemistry more appropriately. So proteins are polypeptide chains of amino acids. An arrangement of amino acids determines different types of proteins. So protein is nothing which is made up of amino acids. Now why I have included protein in gene? Because gene is responsible to produce protein, and protein is nothing but a polypeptide chain of amino acids.